Hi everyone, and welcome to the Chapter 7 lecture on cellular respiration. We'll talk about what cellular respiration is and what it means later in the lecture, but we're going to open with a little thought experiment where I want you to think about what you ate for breakfast today. So maybe you had eggs and bacon, maybe you had some yogurt and fruit, or a bowl of cereal, or something convenient like a protein shake. These foods are all quite different from each other, obviously, but what they have in common is that they provide you with some combination of the three nutritional macromolecules of carbohydrates, lipids, aka fats, and proteins. And remember, the fourth class of macromolecules is nucleic acids, but those are negligible and don't contribute to nutritional value. So we've got our carbs, fats, and proteins. These are the things that fuel you and provide you with energy. How does your body do that? How does your body break down these carbs, lipids, and proteins? Well, in our last chapter, we talked about how these molecules store enormous amounts of chemical potential energy, which can be converted into kinetic energy, or the energy of motion, through your metabolism in order to drive all of your necessary processes. And we also talked about how when we are harvesting that chemical energy through our digestive process, it is necessary to break down those molecules by splitting them open, breaking their chemical bonds, because it is the electrons that house the energy in these molecules. And by breaking those bonds, the energy carried in the electrons can be transferred into a more easily accessible form that can be used for all of your kinetic processes. So all of that should be review from the last chapter lecture. But where we're going to go now is we're going to discuss the overall process for harvesting chemical potential energy from sugar molecules specifically. Everything that you eat, from sugars to fats to carbohydrates to proteins, everything that you eat is broken down in a different way. And it's beyond the scope of our class, and we simply don't have time to talk about how organisms break down all of these different types of molecules. And so for our purposes here, we're going to be limiting the scope of what we talk about by simply discussing the process of breaking down sugar molecules and converting their chemical potential energy into another molecule called ATP, which we've mentioned before as being a fuel molecule for the cell, but we haven't talked about it in detail, so we will do that in this lecture. So cellular respiration is the name for this process. It's the name for harvesting the chemical energy stored in sugar molecules and converting it into another molecule called ATP. This process takes place over the course of 4 different steps, and those 4 steps are: first, glycolysis; second, pyruvate oxidation; third, the citric acid cycle; and fourth and finally, oxidative phosphorylation. So each of these 4 steps represents a series of chemical reactions that gradually break down the sugar and harvest energy from it. Where is this happening inside of your cells? Well, the first step of glycolysis happens inside of the cytoplasm of your cells, which you may remember is the jelly-like substance that fills up the inside of the cell. But the other 3 steps all occur inside of that organelle that we previously talked about called the mitochondria. Pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation all occur inside the mitochondria. And to review, this is what a mitochondrion looks like. It's important to be aware of the different internal features of a mitochondrion as we move forward in this lecture, because we're going to be talking about the different spaces where all of these different processes occur. So mitochondria have 2 separate layers of membranes. They have an outer phospholipid bilayer that makes up the overall egg shape of the organelle. And then inside of that, they have another membrane, which is sort of folded into all of these little curves here. So 2 layers of membranes called the outer and the inner membrane. And there's a specific name for the region in between the 2 membranes. It's called the intermembrane space. And then there's also another name for the region inside the innermost membrane. So the innermost part of the mitochondria is called the matrix. And all the little folds that you see in the inner membrane are called cristae. So keep these things in mind as we're moving forward. We're talking about the different steps in cellular respiration and sugar breakdown that happen in these different regions of the mitochondria. Now it's time to do a little bit more review and recall some concepts that we learned in the last chapter when we talk about the goal of cellular respiration here. So the purpose of cellular respiration is to oxidize compounds in order to extract energy from their chemical bonds. What does that word oxidize mean? Well, in Chapter 6, we learned that there are certain types of reactions called oxidation reduction reactions or redox reactions that involve transferring electrons from one molecule to another. And this is important because remember, it is the electrons that carry the energy. So if you want to transfer energy, you have to transfer electrons. Oxidized is a term that refers to atoms and molecules that lose electrons, and reduced is a term that refers to atoms and molecules that gain electrons. And if that seems like it's a little bit counterintuitive, that reduced refers to a gain of electrons, the reason why it's called reduced is because electrons have a negative charge. And so if you gain more negative charges, overall, the charge of the molecule is reduced because it becomes more negative. That's the origin behind that terminology. So, to summarize, the goal of cellular respiration is to harvest energy from molecules of sugar by extracting electrons from them, because the electrons are the source of the chemical energy that they store. In our last chapter, we did a checkpoint on this particular example of an oxidation reduction reaction, where we saw that there was a sodium ion on the left and a fluorine on the right, and the sodium was being oxidized and the fluorine was being reduced because sodium was losing an electron and fluorine was gaining an electron. This is a very simple and straightforward example of a redox reaction. But the redox reactions that we are going to be observing as we walk through the steps in breaking down the sugar glucose are not going to be as simple as the one that you see here. So in cellular respiration, the loss of an electron is often coupled with the loss of a proton as well. This is for stability purposes because electrons have a negative charge, protons have a positive charge, and the cell can create an increased level of instability if it pulls a negative charge away from a molecule without also pulling away a positive charge. So sometimes it extracts them together. This means that a typical oxidation reduction reaction that occurs in cellular respiration is usually involving the transfer of a hydrogen atom, or rather the equivalent of a hydrogen atom, from one molecule to another. And that's because a hydrogen atom, with hydrogen being the smallest and simplest element on the periodic table, is merely composed of a single proton that represents its nucleus and a single electron located in its orbitals. So if one proton and one electron are extracted from a sugar molecule, that means that the equivalent of a hydrogen atom has been extracted. And these types of redox reactions, when they happen this way, are referred to as dehydrogenations. And the name dehydrogenation is self-explanatory because it is dehydrogenating the molecule that is having its electron and proton extracted. Let's take a look at an example of a redox reaction that dehydrogenates a fatty acid. And now remember, the scope of this lecture is not going to cover how our bodies break down fatty acids. We're going to talk about sugars, but this is just one example that will allow you to clearly visualize what's going on in the redox reactions that are also dehydrogenations. Here we have our modified fatty acid chain, consisting of carbon atoms that are all bonded to hydrogen atoms. And each one of these lines right here represents a covalent bond between the participating atoms. So if we were to redraw some of these lines, in terms of their electrons, this is what they would look like. So we have a carbon atom here that is sharing one electron, and a hydrogen that is sharing another electron, and this pair of electrons that is shared between them represents their covalent bond. In order to extract the energy that is stored in the electrons of that bond, obviously you have to break that bond, and there are certain molecules that we will see later on that are responsible for picking up electrons, such as this molecule called FAD. FAD is something we'll talk about later, but what FAD is capable of doing is dehydrogenating molecules in order to pick up electrons from them and transfer that energy to a different molecule. When this happens, FAD is turned into FADH2 because it effectively removes 2 hydrogen atoms from the molecule, and that means that these carbon atoms' electrons are left without a partner. The solution is that those electrons then get together and they form a new bond with each other, which then turns into a double bond, a covalent bond between those same 2 carbon atoms, which is twice as strong. The electrons that were pulled away by the FAD molecule can then be transferred to some other molecule, and their energy goes with them. So as I said, we're going to focus on sugars, even though the example we just looked at was that of a fatty acid or a lipid. And for a little bit of review, what I want you to do in this checkpoint is tell me what biological macromolecule out of the 4 classifications are sugars the monomer of? Sugars are the building block of which of the 4 biological macromolecules? So this sugar right here, the one that you see in this checkpoint, this is the sugar glucose. And we are going to even further narrow our focus to just talking about how our bodies break down glucose. What you see here is the overall recipe for the breakdown through cellular respiration of a glucose molecule. The chemical formula for a molecule of glucose is, as you see in purple, C6H12O6, which means that within that chemical compound there are 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. So where do you get the glucose from? Well, you eat it. You get it in your diet. That's where that ingredient comes from. The other ingredient that is needed for breaking down this glucose is 6 molecules of oxygen gas. Where do you get that oxygen gas? Well, obviously you inhale it. And this is the purpose of the oxygen gas that you inhale. It is needed to drive your metabolic breakdown of molecules such as glucose. Then in this recipe, we come to an arrow. And what this arrow represents is what all of this stuff on the left side of the arrow gets transformed into over the course of this chemical reaction. So as glucose is broken down with the help of oxygen, it is transformed into 6 molecules of carbon dioxide. What happens to this is you exhale it as a waste product. You also make 6 molecules of water, and that water goes on to become part of the normal volume of water that makes up the majority of your body mass. And then finally, it yields a lot of energy, specifically in the form of that special fuel molecule called ATP. ATP is a very important molecule because it is considered to be the energy currency of the cell. ATP stands for adenosine triphosphate. And adenosine triphosphate is a neat little molecule because although it is very small, it stores a lot of energy for its size, and it stores that energy in a way that is easily accessible to the cell for all of your different kinetic processes. Before we move forward and talk about how ATP does this, I want you to take a closer look at the structure of ATP and tell me what it closely resembles. This looks like a molecule that we've talked about before, and looking at its structure, you should be able to tell me what this molecule of ATP kind of looks like, although it's not exactly the same thing. So, as I said, ATP is the cell's energy currency. Why is ATP the energy currency and not other molecules like sugars, carbs, and fats? Well, that's because sugars, carbs, and fats do store a large amount of chemical potential energy, but they store it in a way that is not easily accessible to the cell for all of its processes. ATP, on the other hand, stores lots of chemical energy in a way that can be used by the cell for any purpose at any time, anywhere. So we can think about sugars, carbs, and fats as being sort of like gold bars or precious jewels or Bitcoin. All of these things hold value, but you can't walk into the grocery store and expect to pay for your groceries with a gold bar. That transaction just won't work. And so we can think about ATP as being sort of like the cold hard cash of the cell. It carries energy and it can be used anywhere for any purpose, unlike the other valuable but less accessible molecules such as sugars, carbs, and fats that you get in your diet. So, how is it that ATP, for being a rather small molecule, stores so much energy in its chemical bonds? Well, if we look at the structure of ATP, we can see that on the left-hand side of the molecule here, there are three phosphate groups, each of which are bonded to the other. And this is the namesake of ATP. It's adenosine triphosphate because there are three phosphates here. We know that the phosphate groups each are negatively charged and that they carry that negative charge on one of their oxygen atoms here. And another thing that we know about negative charges is that while they are attracted to positive charges, they are repelled by other negative charges, kind of like the same end of a magnet. Keeping these three phosphate groups in a covalent bond within the same molecule requires a ton of energy because of the repulsion in between the adjacent negatively charged oxygen atoms. If you have ever held a magnet and tried to force the north pole of the magnet up against the north pole of another magnet, you know that you can physically feel the repulsion between the two, and it's almost impossible to bring them together. The closer you bring them, the harder you have to push in order to keep them there. And so it's the same principle here. With these oxygen atoms having a negative charge, and with those negative charges being in such close proximity, the bonds that hold the phosphate groups together contain a lot of energy and a lot of effort to keep them there in that stable location. This is where the energy is ultimately stored in an ATP molecule. When the cell needs to access that energy in order to operate a chemical reaction that requires it, it will break the bond to the third phosphate group, and by allowing that bond to break, the phosphate group flies off and its chemical potential energy that was previously being used to hold that phosphate group there is now released for some other purpose. Once that happens, the adenosine triphosphate molecule is actually no longer adenosine triphosphate, it is now adenosine diphosphate because it has lost one of its three phosphate groups and it only has two, which is why it is diphosphate. We can kind of think about ATP as being the fully charged version of the molecule and adenosine diphosphate, ADP, as being the uncharged version of it. But the beauty of these molecules is that they can be charged back up again, and the only thing that the cell has to do is attach another phosphate group. And the energy that it gets to do that comes from your nutritional molecules that you get in your diet. So really, the goal of metabolism is to take these more long-term potential energy molecules like carbs, lipids, and proteins, convert them into this more short-term and accessible potential energy in the form of ATP molecules, And then those ATP molecules can be burned and turned into kinetic energy that is used to power all of an organism's processes. So let's take a deep breath for a second, take that all in, and look back at this metabolic map that I showed you guys in the previous lecture when we were talking about enzymes. But I want to zoom in on one particular part of this metabolic map and show you what we're going to be talking about for the rest of this lecture now that we've sort of laid the foundations and introduced the concepts that we're talking about. We are going to be focusing on the conversion of simple sugar molecules, specifically glucose, through a number of chemical reactions powered by enzymes that will eventually lead to the production of energy in the form of ATP and, of course, heat, as we discussed previously. So we're going to follow this path from sugars all the way through the 4 different reactions that we talked about previously, which are glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. Over the course of these 4 steps, what we are going to see is that oxidizing one single glucose molecule through this process called aerobic cellular respiration produces an average of 38 molecules of ATP per glucose molecule that runs through these chemical steps. So we're going to walk through these different stages one by one, starting with glycolysis, and we are going to follow a glucose molecule through its complete breakdown into ATP molecules. Remember that glycolysis is the only one of the 4 steps that occurs in the cytoplasm of the cell. So we have not yet entered the mitochondria. Instead, the glucose molecule starts in the cytoplasm, gets broken down partially, and then its component parts move into the mitochondria for the rest of the process. Let's take a look at what happens during glycolysis. So glycolysis starts with a glucose molecule, and that's what you see represented here. It's a little bit different look at a glucose molecule that we've seen previously because most of the carbon atoms have been made invisible here, but trust the fact that they are still here. Glucose will be broken down through glycolysis into 2 molecules of a different chemical called pyruvic acid, or in other words, pyruvate. This is what pyruvic acid looks like. And as you can see, it kind of looks like half of a glucose molecule. And indeed, the name glycolysis, what this literally means is glyco refers to sugar, and lysis refers to break. So glycolysis literally means to break the sugar in half. And that's what is achieved in this process. The glucose sugar is broken in half, and the name for each half of the glucose is pyruvic acid. When these bonds are broken, some of the chemical energy is harvested. Enough chemical energy is harvested to recharge 2 molecules of ADP, adenosine diphosphate, back up into their fully charged forms of ATP, adenosine triphosphate. This is not the only way that chemical energy is harvested, however, because something else happens in glycolysis as well, and that is a dehydrogenation reaction. Electrons and protons are also harvested from the glucose molecule when its bonds are broken by a special molecule called NAD+, which stands for nicotinamide adenine diphosphate, but I don't want you to know that. We're going to look at the structure of NAD+ in just a moment, but for right now, all you need to know is that NAD+ picks up electrons and hydrogen from the broken glucose molecule twice. This happens with 2 separate NAD molecules, and thereby they are converted into their charged up form of NADH. You can see how the dehydrogenation has taken place, indicated by the fact that there is now a hydrogen atom on the NAD+ molecule. So at the end of glycolysis, the energy harvesting process has yielded 2 molecules of ATP, as well as 2 molecules of this thing called NADH. And as promised, here's a look at the structure of NADH, or nicotinamide adenine diphosphate. Again, don't have to know that, only have to know it as NAD. If we look closely at the structure of NAD+, which is shown on the left-hand side here, versus NADH, which is shown on the right-hand side here, then the only difference between NAD+ and NADH can be seen up here, where we see that instead of there being one hydrogen atom located in this area of the compound, now there are 2 hydrogen atoms. And so, again, NAD is responsible for performing these dehydrogenations that pick up protons in combination with electrons, and it carries that hydrogen that it picks up right here. So what is going to happen to the energy that has been collected by the NAD? Well, we're going to see that these NAD molecules are going to come back into the picture in a later step of the cellular respiration process, and their energy is going to be converted into molecules of ATP as well. Before we move on to the next step of the process, let's take a look back at the overall recipe, or the overall chemical equation for this cellular respiration thing, which, remember, is glucose plus some oxygen together yield carbon dioxide, water, and energy in the form of ATP. Out of each component in this overall chemical equation, which ones did we see featured during glycolysis? Out of the ones on the ingredient side, which ones did we see active? And out of the ones on the product side, which ones did we see created? So what we saw in glycolysis is that the molecule of glucose was broken down and split in half into 2 molecules of pyruvic acid, or in other words, pyruvate. Now we're going to move on to the second step of the process, pyruvate oxidation. And at this point, we're going to move inside of the mitochondrion here. Remember, glycolysis takes place in the cytoplasm, but pyruvate oxidation occurs when the pyruvate molecules move into the intermembrane space. So they have passed through the outer membrane of the mitochondrion, but not through to the matrix area. They're only existing in between the 2 layers of membrane. So let's see what happens during pyruvate oxidation, which to me is the most self-explanatory step of the process because it tells you exactly what is going on during the chemical reaction, which is the oxidation of pyruvate. Remember, oxidation means to lose electrons, and so we are going to see the pyruvic acid, or the pyruvate, lose electrons in this step as energy is harvested from it. What happens during pyruvate oxidation is the pyruvic acids are further broken down into another molecule called acetyl-CoA. This is what acetyl-CoA looks like, and as you can see, it is similar in structure to the pyruvic acid, but it is missing some stuff, because in the process of breaking down this pyruvic acid, a molecule of carbon dioxide is removed from the pyruvic acid and released as a waste product. And remember, this is exhaled by us. Also during this process, another one of those molecules of NAD comes along and dehydrogenates the pyruvic acid and picks up another pair of electrons from it and turns into NADH. Now it's important to remember that what you see on the screen here represents the breakdown of a single pyruvic acid. One pyruvic acid turns into one acetyl-CoA, and in the process, one molecule of CO2 is released and one NAD+ picks up a hydrogen atom. However, for every glucose molecule that starts the process off and runs through glycolysis, remember that 2 pyruvic acids are created because a pyruvic acid is effectively 1/2 of a glucose molecule. So the glucose gets split in the first step, yields 2 pyruvic acids, and then each of these pyruvic acids goes through pyruvate oxidation. So if we go to quantify how much energy has now been harvested at the end of the pyruvate oxidation step, we still have a mere 2 ATP, both of which were created during glycolysis. And now we have a total of 4 NADH molecules that have dehydrogenated and extracted electrons from glucose and its derivative molecules, because remember, this whole process happens twice. So we don't count just one NAD molecule. We have to consider that there are actually 2 NAD molecules created, one per pyruvic acid that comes out of glycolysis. So 2 NADH molecules were created in glycolysis, 2 NADH molecules were created in pyruvate oxidation. That brings us to a total of 4 NADH molecules collected so far. Now, just like we did in the previous checkpoint, tell me in this checkpoint which components of the overall chemical equation for cellular respiration did we see active in pyruvate oxidation? Were there any that were consumed, and were there any that were produced? So now we're moving on to the third step of the process, which is called the citric acid cycle. And the citric acid cycle will move us even further into the mitochondria, because while pyruvate oxidation took place in the intermembrane space between the two layers of membranes, the citric acid cycle, or the Krebs cycle, as it is sometimes called, occurs in the matrix, which is the area inside the innermost membrane of the mitochondria. The citric acid cycle is named for the fact that it is cyclical in nature, and it involves the chemical compound citric acid. The process starts with the acetyl-CoA molecule, which is coming out of pyruvate oxidation, and yet it still has more energy to be harvested, and that's what's going to take place in the citric acid cycle. The citric acid cycle incorporates the acetyl-CoA molecule into an existing molecule of oxaloacetic acid, but I don't want you to know the names of these chemical compounds that you see in the cycle here. This is just for your visual inspection. What I want you to know is that the citric acid cycle brings in the acetyl-CoA, and then over the course of 8 separate chemical reactions, it breaks down the acetyl-CoA gradually, and in the process, it releases 2 molecules of carbon dioxide per molecule of acetyl-CoA that starts this process off. In the process, more electrons are also harvested by these molecules called NAD. More electrons are harvested by a separate electron harvesting molecule called FAD, which we saw in our example that we talked about at the beginning of the lecture. And then finally, 1 molecule of ATP is created or recharged from the uncharged version of ADP. So as you can see, this was a pretty productive process here. And again, recall that this represents how many molecules of NADH, FADH2, ATP, and carbon dioxide are generated from the breakdown of a single acetyl-CoA molecule. But again, remember that because we started with 1 glucose molecule, which was broken in half into 2 pyruvic acids, and each of those pyruvic acids became an acetyl-CoA, this whole process happens twice for every glucose molecule that is broken down. So if we were to quantify how the energy harvest process is going at this point, we know that 2 molecules of ATP were created during glycolysis. We also know that a molecule of ATP is created for every acetyl-CoA that is broken down in the citric acid cycle. And because this happens twice for each acetyl-CoA that comes out of pyruvate oxidation, 4 ATP have been created at this point. We also know that 2 molecules of NADH were created in glycolysis. We know that a net total of 2 molecules of NADH were created in pyruvate oxidation. And then finally, we see that 3 molecules of NADH are created per acetyl-CoA. And because there's 2 acetyl-CoAs, that means that there's really a total of 6 NADH molecules created here. So 2 from glycolysis, 2 from pyruvate oxidation, and 6 from the citric acid cycle means that a grand total of 10 NADH molecules have harvested electrons from glucose and its derivatives at this point. We also see that some molecules of FADH2 have been created, specifically 1 per acetyl-CoA, meaning that there's a grand total of 2 per glucose molecule that starts the process off. And if you think about it, the citric acid cycle is step 3 of 4 of this process. We only have one more step to go, and yet we've only created 4 ATP molecules at this point. I told you at the beginning of the process that an average of 38 were going to be created per glucose molecule. So we can predict that there's going to be an absolute landslide of ATP coming in this 4th and final step. And where the energy to make that ATP comes from is in all of these molecules of NADH and FADH2, which are carrying electrons that have been harvested from glucose and its derivatives. But before we move on to talking about the 4th and final step of cellular respiration, once again, I want you to tell me, out of these components in the overall equation for cellular respiration, which ones did you see coming into play during the citric acid cycle? Was anything from the overall equation consumed, and was anything produced? So we've now arrived at the 4th and final step of cellular respiration, which is oxidative phosphorylation. Oxidative phosphorylation, again, occurs inside of the mitochondria, and it happens specifically at the inner mitochondrial membrane. And we don't say that it happens either in the matrix or the intermembrane space because it's actually going to be occurring across the membrane itself. And here's what that process looks like. So just to get our bearings here with this diagram and what it's showing us, we can see the phospholipid bilayer here. And we can also see these big blue blobs, which are proteins embedded in the membrane. This phospholipid bilayer is the inner mitochondrial membrane. So on one side of it is the matrix, or the innermost region, and on the other side is the intermembrane space between the outer and inner membrane. So as we said, we have only made, up until this point, 4 molecules of ATP out of that 1 molecule of glucose that started the process off. So we're kind of measly in terms of ATP, but what we have made is a large amount of these NADH and FADH2 molecules, which are carrying electrons and will now move into oxidative phosphorylation, where they will dump their electrons off in order to convert that energy into ATP. So the NADH and FADH2 molecules that were made in the first 3 steps of cellular respiration will travel to the inner mitochondrial membrane, and these NADH and FADH2 molecules, which are each carrying a set of electrons that were extracted, are going to transfer those electrons to the proteins embedded in the inner mitochondrial membrane, which are going to be responsible for ultimately generating the ATP. So let's take a look at how this happens. The name of the first protein involved in this process is called NADH dehydrogenase. This is an enzyme, and it is named for its function because what it does is it dehydrogenates the NADH. NADH is carrying those electrons, and NADH dehydrogenase, the protein, is going to take those electrons away from it. When it takes those electrons away from the NADH, first of all, returns to its original form of NAD+, and this NAD+ can then go back to a previous step and pick up more electrons. These electrons here that have been transferred to the protein, their energy is specifically used to pump hydrogen ions, or in other words, protons, from the matrix into the intermembrane space. There is a pretty much constant supply of protons hanging around in the mitochondrial matrix. And although they do not want to move in this direction, they can be actively transported by using the energy of the electrons to move them from the matrix side to the intermembrane space side. Why would the cell want to do this? Why is it wasting its energy moving protons from one side of the membrane to the other? Well, we'll see why in just a second. But before we get to that, let's take a look at the fate of these electrons here that have been transferred from the NADH now to the NADH dehydrogenase, and we can see that where they're going to go next is onto this little molecule right here called ubiquinone. Ubiquinone is what we can kind of think about as being an electron shuttle. Its whole responsibility is to pick up electrons from this protein and move them to this protein right here. This protein is called the BC1 complex. And when the BC1 complex gathers those electrons from ubiquinone, it too uses the energy in those electrons to actively pump another proton from the matrix of the mitochondria to the intermembrane space here. So now we have an even larger quantity of protons accumulating in the intermembrane space. After this happens, the electrons are transferred to another electron shuttle called cytochrome C. And cytochrome C's entire job is, like ubiquinone, to move the electrons between the BC1 complex and the third and final protein here called the cytochrome oxidase complex. Again, no surprises here. The cytochrome oxidase complex will use those electrons to actively fuel the pumping of hydrogen ions or protons from the mitochondrial matrix into the intermembrane space. And just so you guys know, hydrogen ion and proton, those two words are used synonymously in this context. A hydrogen ion is the same thing as a proton. When this pumping action is complete, the electrons are spent of their energy. They have no more energy left to give, and so the cytochrome oxidase complex just wants to get rid of them. It will dump those electrons onto a molecule of oxygen. Again, this oxygen was one of the ingredients in the overall process for cellular respiration, and here's where we see it come into play. The oxygen is the final acceptor of these spent and no more energetic electrons after they have finished pumping all of these protons from the matrix to the intermembrane space. When the electrons land on this molecule of oxygen, they combine also with some protons or hydrogen ions, which again, there's a large concentration of those in the matrix. And the combination of the electrons, the H's, and the O's turns this into water. Remember that water is another one of the outputs of the cellular respiration overall chemical equation. One thing that we want to note here before we move on is what happens to the FADH2s. Because remember, although there are only two of these guys, they are carrying some electrons from the citric acid cycle. We can see that unlike the NADH molecules, which unload their electrons onto NADH dehydrogenase, the FADH2 molecules unload their electrons directly onto ubiquinone instead of the NADH dehydrogenase. So they effectively sort of get on the bus one step later than the NADH dehydrogenase electrons, and therefore these electrons pump fewer protons ultimately than those from NADH because they're not involved with NADH dehydrogenase protein at all. So what has been accomplished after all of this is complete? We now have a very high concentration of protons in the intermembrane space and a relatively lower concentration of protons or hydrogen ions in the mitochondrial matrix. And based upon what we've learned about diffusion and passive transport, these highly concentrated protons are going to want to move to an area where they are less concentrated because they want to spread out. They accomplish this spreading out motion by diffusing back across the membrane and into the matrix through a special enzyme called ATP synthase, which is what you see right here. ATP synthase is a transmembrane protein that crosses the membrane, and it acts as a channel for these hydrogen ions to rush back to the original side of the mitochondrial matrix because that's the direction that diffusion dictates that they want to go. Now, ATP synthase is a very cool enzyme. Its name directly implies what it does, which is that it synthesizes or makes ATP, but the way that it does it is very unique. ATP synthase acts like a rotary motor, meaning that as these protons flow through the channel of the ATP synthase, it causes the ATP synthase enzyme to physically rotate. Much like a water wheel, the flow of the protons is like the water that pushes the wheel around. And as the enzyme rotates, that rotational energy is used to drive the ATP creation. Specifically, for every 10 protons that run through this water wheel of an enzyme, 3 ATP are created. Or in other words, it takes 3.33 protons to make each ATP molecule. This right here, what you're seeing, might not look like much, but it is really cool because this is actually looking through a microscope at that ATP synthase enzyme that has been attached to some fluorescent dyes. And what you're actually seeing here is the enzyme rotating as the hydrogen ions move through it. So this is ATP synthase in action right here under the microscope, which is really cool. So on average, when you count up all of the protons that are pumped by the action of the NADH and the FADH electrons, and all of the ATP that they are able to make by rushing back through ATP synthase, an average of 34 ATP molecules can be generated during this step per glucose molecule that is broken down in the first 3 steps. That's where the rest of our 38 ATP come from. Only 4 come from the first 3 steps, but 34 are made in this last step of oxidative phosphorylation. So knowing what you know now about how ATP synthase works, I want you to tell me how many molecules of ATP would be produced if 50 protons flowed through the ATP synthase enzymes. And next, I want you to tell me whether the electrons that are carried by NADH or FADH2 are more productive in terms of generating ATP. And why would that be the case? And I'll give you the clue. The answer is not NADH because there are more NADH molecules there. I want you to tell me on a molecule for molecule basis which one is more productive, an NADH or an FADH2, and why. So at this point, we have walked through the complete breakdown of a glucose molecule and seen how it has gone through the extraction of its electrons and the creation of large amounts of these ATP fuel molecules from its chemical energy. There's only one last topic that we have to cover, and that is an alternative process to cellular respiration as we have discussed it. The steps that we just walked through are called aerobic cellular respiration. And they're called aerobic cellular respiration because the process requires oxygen or air, hence aerobic. There is another process that many organisms are able to perform in the absence of oxygen, which is called anaerobic cellular respiration. And this process can occur as a sort of a backup or a plan B if oxygen is not available in order to produce a small amount of energy, nowhere near 38 ATP, but enough energy to keep an organism going. Interestingly, glycolysis is the first step for both of these processes. Anaerobic respiration starts with glycolysis just like aerobic respiration does. But what determines whether glycolysis will lead into pyruvate oxidation, the citric acid cycle, and finally the electron transport chain of oxidative phosphorylation is whether or not there is oxygen available. If the cell is in an oxygen-rich environment, then glycolysis will lead into the steps that we just talked about. However, if oxygen is not available, then glycolysis will instead lead into anaerobic respiration by segueing into a process called fermentation. And glycolysis was originally discovered by scientists not because it was part of aerobic respiration, not because people wanted to know more about the process by which our cells acquire energy, but because they wanted to learn more about fermentation, and they were studying how to ferment better wine. So that's an interesting fact. So let's talk about why this process that we just talked about, known as aerobic cellular respiration, cannot occur in the absence of oxygen. We just watched the electrons that were carried by NADH and FADH2 run through a series of transports from protein to protein and eventually land on a molecule of oxygen there. Now, oxygen is usually available to our cells, except when we are in really, really hard aerobic exercise states. So what happens when that oxygen gets taken away? Well, as you can imagine, if that oxygen is not present at the end of this chain, the cytochrome oxidase complex has nowhere to drop its useless electrons. If the cytochrome oxidase complex can't get rid of its electrons, then cytochrome C cannot deliver new electrons to the cytochrome oxidase complex because it is already full of electrons. If cytochrome C is full of electrons, then the BC1 complex can't pass off its electrons to cytochrome C, and likewise ubiquinone can't pass off its electrons to the BC1 complex. If we follow this logic back to its original starting point, then eventually we're going to get to a point where everything is so backed up that NADH and FADH2 no longer can unload their electrons onto these proteins here. And so therefore NADH and FADH2 can never be recharged back into the NADH and FAD form. So here's what this looks like in a graphical format. If oxygen is absent, then oxygen cannot be used as the final collector of those electrons at the end of the third step. If that is the case, then NADH can no longer unload its electrons on the final step and therefore be recharged to NAD+. It is necessary for these NAD+ molecules to be around because they are needed in previous steps such as glycolysis to pick up the electrons. And the cell doesn't have an infinite supply of NAD+. It needs to have this cyclical nature where the NADs pick up electrons, travel to the last step, dump them off, and then travel back to the beginning and pick them up. If you take that away, of course, the system gets backed up to the point where even glycolysis can't run because there's nothing to collect the electrons from the glucose molecule. Now, here's the breakdown of ATP that we saw happening in all of these different steps. We know that glycolysis only produced a grand total of 2 ATP molecules, and that out of the last 3 steps put together, we got another 36. Without oxygen, and when the chain gets really backed up, there is not enough NAD around to run the last 3 steps of the process, and so there is no way for the cell to access those 36 ATP. However, there is a way for the cell to continue running glycolysis if it can rapidly regenerate just a small amount of NAD plus molecules that are needed to collect electrons from glucose in this initial step. And that is the purpose of fermentation. What fermentation does is it allows the NADH molecules that pick up electrons from glycolysis to dump those electrons off on some carbon-based molecule rather than oxygen gas. And this allows NAD plus molecules to be regenerated so that glycolysis can continue to run and generate 2 ATP per glucose, which is completely measly, but it is enough to keep the cell going. So look at this in graphical format. Glycolysis runs like normal. NAD plus molecules harvest electrons and turn into NADH. And then if there's no oxygen available and the system is backed up, then as a last resort, fermentation offers an alternative pathway for NADH molecules to dump those electrons off on some sort of an organic molecule so that there are enough NADs that can go back and allow glycolysis to run. This may seem like a lot of work for only 2 ATP that come out of the process, but as I said, this takes place under emergency conditions, and 2 ATP, while not sufficient for long periods of time, can be enough to keep the cell going under low oxygen conditions. Different types of organisms end up dumping the electrons on different molecules, which are the products of fermentation. Alcohol fermentation is performed by yeast and some bacteria, and so ethanol is the final product of that type of fermentation performed by those species. Lactic acid fermentation is performed by animals such as ourselves, as well as some other bacteria. Lactic acid fermentation is what happens to the cells in your muscles when you're exercising really, really hard and you're going so hard that your body can no longer supply enough oxygen to your cells at the rate needed to support your activity level. And so with that lack of oxygen, your cells kick into fermentation gear and they start to produce lactic acid instead. And this lactic acid is thought to be a major component of what causes people to become sore after a hard workout. Now we have just one more checkpoint at the end of this lecture to finish it off. Let's say you are a brewer who uses yeast to produce alcohol. How would you alter the yeast's environment in order to make this happen and force them to produce alcohol? When you're finished with this checkpoint, then you are finished with the whole lecture.